my name is Genèvive Pearce. My pronouns are she, her. I'm a biology student at Brown University, and here's my honors project, Lactic Acidosis and pH Balance in Breast Cancer. So, as of until more recently, we only really focused on studying the cancer cells itself. So if we look at the seed and soil analogy, this would be more just looking at the cancer cells itself. Except when we have breast cancer, we also have the tumor microenvironment. So that's the environment around it. So it's like how the soil affects how the seed grows. And that's more what my project's gonna be looking at the different conditions that are present in the soil or the tumor microenvironment. So different breast cancer. So breast cancer isn't just like one thing. It's divided into different subtypes. And this is dependent on which receptors they have. So we can have two different, all the same, or none. And this just affects multiple things in the cancer progression, like aggressiveness, but also what the cancers like for their metabolism. So that's just basically what they like to use for their energy. So our luminal A, which is the least aggressive, they prefer this oxidative phosphorylation, which is the PDHA1 enzyme that they use. But our HER2 positive and our triple negative breast cancer, which is the most aggressive that we have, they prefer anaerobic glycolysis, which is in the presence of no oxygen. And this triple negative also, it possesses a unique feature that it's able to switch back between these different metabolisms dependent on their needs, which makes them even more aggressive. So if we look at this metabolic pathway, we also see that there is a lactate byproduct. And as of until more recently, researchers kind of ignored this. They just thought it was a simple byproduct, but actually it's really involved in the tumor microenvironment as it decreases pH and can have effects on its cancer progression. Additionally, cancer cells, they grow in these really intense conditions, like stressful conditions, and they need to have ways to be able to to survive and overcome that. So they have previously known shuttles that can shuttle that pH and lactate, but additionally, they tried targeting that, but there was some toxicity involved, which is why my project is looking at ectopic ATP synthase. So typically, ATP synthase is only present in the mitochondria, but it's been found that in certain rapidly dividing cells, like breast cancer cells, it's found in the plasma membrane known as ectopic expression, and not found in just simple breast cancer cells. So if this was involved in that pH regulation, this would be a really excellent therapeutic target, as we can minimize toxicity by just targeting those cancer cells. So my methods that I did is that I did cancer cells and we grow them in flasks. So typically we have just a normal media, but what I did is I manipulated that media to do different conditions. So I had an acidosis condition, which is just low pH and no lactate. I had a lactosis, which is neutral pH and a high lactate levels. I had lactic acidosis, which is low pH and high lactate and a control. And doing all these conditions is really important to differentiate if it was the lactate or the pH or a combination of both. So I grew my cells in those conditions, and then I did protein extractions, and I quantified, so I measured how much protein of those different enzymes were there. So we could, by looking at the enzymes and how much, we could see which pathway they're going through. So for MCF7, which is our luminal A, we found that when we grew in those lactate high conditions, the enzymes that are responsible for that is oxidative phosphorylation, what they like. There was a shift. So they really didn't like these byproducts, and there was just a shift back to what they typically like. For HER2 positive, which is here, that preferentially likes anaerobic glycolysis, when we added more of those byproducts, their growth kind of slowed down and there was a decrease in expression. So that just showed that by adding more byproducts, their machinery is kind of overwhelmed. They couldn't keep growing, so they just more slowed down. And then for the triple negative breast cancer, which can go back and forth, we found that it could go back and forth. So you can see the expression is very varied, and that's because depending on the conditions, they could switch their metabolism to survive, and they kept growing well. So the future directions is that I want to look, so I found that there's difference in like growth, how well they could grow. So I want to do functional assays to see how well they can generate energy, and then also how well they can like divide and migrate. So that would be really interesting to see in those conditions. And then also I want to look at how inhibiting this ectopic ATP synthase to see if it prevents like their growth or their survival. Thanks.